You will spend roughly a third of your entire life doing something science still cannot fully explain, lying unconscious, paralyzed, and hallucinating for no reason anyone has definitively proven. That's sleep, and it's only the first of 200 facts tonight about the body you've lived inside your whole life without ever really understanding it. We'll cover the gut that runs its own nervous system independent of your brain, home to more neurons than a cat's entire brain. The phantom pain amputees feel in limbs that no longer exist. The immune cells that somehow remember an infection from decades before you were born. And the 98% of your DNA that doesn't code for anything scientists can identify a clear purpose for. We'll sit for a while with the single hardest unanswered question in all of science, why there's a felt experience of being you at all, and end at the quiet cellular clock ticking, right now, at the tip of every chromosome in your body. Some of tonight's facts have tidy explanations. A surprising number don't. Let your own body, the one thing you've never actually had to think about to keep running, carry you gently towards sleep. Every animal with a brain complex enough to study sleeps in some form, from fruit flies to blue whales, and yet, after more than a century of dedicated research, scientists still cannot fully explain why sleep evolved at all. A genuinely strange gap in a science that has otherwise mapped nearly every other basic biological process in exhaustive detail. The most basic argument for why sleep should exist is also, on its surface, the hardest to reconcile with how dangerous sleep actually is. An unconscious animal cannot watch for predators, cannot eat, and cannot reproduce, meaning evolution should, in theory, have found some way to eliminate or drastically shrink such a costly behavior if a lighter alternative were available. Instead, sleep has persisted across roughly 700 million years of animal evolution in some form, appearing even in creatures as simple as jellyfish, which lack a centralized brain entirely, a finding published in 2017 by researchers studying the upside-down jellyfish species Cassiopeia, which was shown to enter a measurably reduced, sleep-like state at night, suggesting whatever sleep actually accomplishes is important enough that evolution has refused to abandon it even in animals with the simplest nervous systems on record. The leading scientific explanation, called the synaptic homeostasis hypothesis, proposed by neuroscientists Giulio Tononi and Chiara Cirelli beginning in 2003, suggests sleep exists primarily to let the brain downscale, weakening the countless synaptic connections strengthened during a day of learning and sensory input, preventing the brain from becoming saturated and preserving its capacity to keep learning the next day. A competing and equally serious hypothesis focuses not on the brain's synapses but on its plumbing, pointing to the glymphatic system, a network of channels discovered and formally named only in 2012 by neuroscientist Maiken Nedergaard's research team, which flushes metabolic waste, including a protein called beta-amyloid linked to Alzheimer's disease, out of the brain considerably more efficiently during sleep than during waking hours. Neither hypothesis has been definitively confirmed as the primary reason sleep evolved, and most sleep researchers today believe sleep likely serves several of these functions simultaneously rather than any single one. Sleep itself is not a single uniform state, but cycles through distinct stages roughly every 90 minutes throughout the night. And one of these stages, rapid eye movement sleep, or REM, presents its own long-standing puzzle. During REM sleep, the brain becomes almost as electrically active as it is while fully awake, while the body simultaneously enters a state of near-total paralysis called REM atonia that prevents the sleeper from physically acting out the vivid dreams typically occurring during this stage. The specific neural mechanism producing this paralysis was only mapped in detail through animal studies conducted at Stanford University and published in 2016, identifying a small cluster of neurons in the brainstem responsible for actively suppressing muscle movement during REM sleep. And researchers still debate why the brain evolved such an elaborate energy-intensive paralysis system, rather than simply reducing the intensity of the dreaming itself. A rare and severe disorder called REM sleep behavior disorder offers a genuinely unsettling real-world glimpse of what happens when this paralysis system fails. First formally described in a 1986 medical paper, the condition causes sufficient sufferers to physically act out their dreams, sometimes violently, punching, kicking, or falling out of bed during an episode they have no memory of upon waking. What makes the disorder scientifically significant well beyond its immediate symptoms is a striking statistical pattern identified through long-term follow-up studies published beginning in the 1990s and refined through the 2010s. A majority of patients diagnosed with REM sleep behavior disorder later go on to develop a neurodegenerative condition, most commonly Parkinson's disease, sometimes more than a decade after the sleep disorder first appeared, making the paralysis failure one of the earliest known warning signs of a disease process that would otherwise remain undetectable for years. Total sleep deprivation experiments, conducted primarily on animals for ethical reasons, have produced results serious enough to settle any remaining doubt about sleep's basic necessity, even without a full explanation of its underlying mechanism. Rats kept in a specialized apparatus that prevented sleep entirely, in a series of experiments conducted at the University of Chicago, beginning in the 1980s by researcher Alan Rechtschaffen, reliably died within 2 to 3 weeks, developing skin lesions, uncontrolled weight loss despite eating more than usual, and a complete collapse of body temperature regulation, even though no single organ failure could ever be pinpointed as the direct cause of death, leaving researchers able to say with total confidence that sleep is not optional for survival, while still not being able to say precisely which of sleep's many functions, if permanently withheld, is the one that actually kills. The record for the longest documented period of voluntary human sleep deprivation belongs to Randy Gardner, a California high school student who in December 1963 at age 17 stayed awake for 11 days and 25 minutes as part of a science fair project, closely monitored throughout by Stanford sleep researcher William Dement. Gardner experienced progressively severe cognitive effects, including memory lapses, irritability, and in the later days, brief hallucinations and paranoid thinking, yet recovered fully after a single extended sleep of about 14 hours, a case that remains frequently cited in sleep research precisely because it demonstrated both how far human wakefulness can be pushed and how completely the brain can recover, at least from one such extreme episode, once normal sleep resumes. Once upon a time. Before we go on, if lying back and letting 200 facts about your own body wash over you sounds like your kind of evening, this channel does exactly that on a regular schedule, and subscribing costs nothing beyond making sure the next one finds you. Now back to the strange architecture of a single night's sleep. Certain animals have evolved a genuinely remarkable workaround to sleep's most basic vulnerability, the inability to stay alert while unconscious. Dolphins, along with several other marine mammals and some migratory birds, practice what's called unihemispheric slow-wave sleep, in which one half of the brain sleeps while the other half remains awake and alert, with one eye typically staying open on the side corresponding to the waking hemisphere. This allows a bottlenose dolphin to continue swimming, surfacing to breathe, and watching for predators while still obtaining the rest its brain requires. And researchers studying frigate birds, which can remain airborne continuously for more than 2 months during transoceanic flights, published findings in 2016 confirming the birds use the same unihemispheric technique mid-flight, sleeping in short bursts of only seconds at a time. While one half of the brain briefly shuts down, a solution so effective that some sleep scientists have proposed studying it more closely for insight into what sleep's essential, non-negotiable function actually is, since these animals appear able to obtain whatever that function provides in far smaller and stranger doses than humans require. Sleep's timing, distinct from its underlying function, is governed by an internal circadian clock whose physical location remained a genuine mystery until 1972, when two independent research teams identified the suprachiasmatic nucleus, a cluster of roughly 20,000 neurons sitting just above where the optic nerves cross inside the brain, as the body's master pacemaker. Direct evidence that this tiny structure alone controls the timing of sleep and wakefulness came from a striking series of experiments in which researchers surgically transplanted a suprachiasmatic nucleus from one hamster into another hamster whose own had been destroyed, and found the recipient animal's sleep-wake cycle shifted to match the donor's specific rhythm rather than its own original one. Direct proof the structure itself, rather than any broader brain region, sets the clock. French geologist Michel Siffre provided one of the most extreme human demonstrations of this. Internal clock's independence from the outside world, spending 205 days alone in a cave in Texas in 1972, with no natural light clocks or calendars of any kind, and found his body naturally settled into a sleep-wake cycle slightly longer than 24 hours. Direct evidence that the human circadian rhythm runs on its own internal timing, even when completely isolated from the sun, though it still requires daily light exposure to stay precisely synchronized with the actual 24-hour day, a process called entrainment that remains only partially understood at the level of exactly how much and what wavelength of light is required to reliably reset the clock in every individual. A far briefer and considerably more dangerous lapse into sleep, called microsleep, involves an involuntary episode lasting anywhere from a fraction of a second to about 30 seconds, during which a person's brain briefly enters sleep-like activity patterns without the person necessarily even realizing it happened, eyes sometimes remaining open throughout. Researchers studying microsleep through EEG monitoring have found it occurs considerably more often during states of severe fatigue or sleep deprivation, and it has been directly implicated as a contributing factor in a substantial share of serious vehicle accidents, according to figures compiled by the National Highway Traffic Safety Administration. Precisely because a driver can experience a microsleep episode, briefly lose full awareness of the road, and regain consciousness with no memory the lapse ever happened at all, a genuinely unsettling illustration of how porous the boundary between full wakefulness and sleep can become under the right conditions, and one researchers still cannot fully predict at the individual level, since two equally fatigued people can show measurably different susceptibility to microsleep episodes under otherwise identical conditions. Sleepwalking, formerly called somnambulism, belongs to a broader category of sleep disturbances called parasomnias, and, genuinely counterintuitively, occurs not during the vivid, dream-rich REM stage most people associate with active mental experience, but during deep non-REM sleep, the stage typically associated with the least mental activity of the entire night. Researchers using EEG monitoring have found the sleepwalking brain exists in a genuinely unusual intermediate state with the motor cortex, responsible for physical movement, showing wake activity levels, while the prefrontal cortex, responsible for judgment, planning, and full conscious awareness, remains in a deep sleep pattern, effectively producing a body capable of walking, and in rare documented cases performing considerably more complex behaviors, while the part of the brain that would normally supervise and remember those actions stays largely offline. A 2018 study, published by sleep researcher Francesca Ciclari's team, using high-density EEG to map sleepwalking episodes in real time, confirmed this specific split activity pattern directly, though what triggers any individual episode on any given night, and why the condition runs so strongly in families, with twin studies suggesting a substantial genetic component, while still requiring some additional environmental trigger like sleep deprivation or fever to actually produce an episode, remains only partially understood. Within a single night's sleep, brain activity recorded through EEG monitoring reveals a further layer of unexplained structure. Brief, distinctive bursts of fast neural oscillation called sleep spindles, along with sharp voltage spikes called K-complexes, both occurring primarily during the deeper stages of non-REM sleep, and both first formally documented in EEG recordings published during the 1930s. Researchers studying sleep spindles more closely in recent decades have found their frequency correlates measurably with a person's performance on memory tasks the following day, and a widely cited 2013 study led by researcher Bryce Mander found that people who naturally produce more sleep spindles show better preserved memory function into older age, suggesting spindles may play a direct role in consolidating memories overnight, though the precise mechanical process by which a brief burst of electrical oscillation actually transfers information into durable long-term storage remains an active and unresolved subject of ongoing neuroscience research. Sigmund Freud's 1899 book, The Interpretation of Dreams, proposed that dreams represent disguised wish fulfillment, a theory that dominated popular thinking about dreaming for much of the 20th century, despite never being validated through any rigorous scientific method, and modern dream researchers have largely moved away from Freud's specific framework while still finding themselves unable to agree on a replacement theory that fully explains why the sleeping brain generates the vivid, often bizarre narrative experiences most people recall at least fragments of upon waking. One of the more influential modern theories, called the threat simulation theory, proposed by Finnish cognitive scientist Antti Revonsuo in 2000, suggests dreaming evolved as a kind of offline rehearsal space, allowing the brain to simulate dangerous or threatening scenarios in a low-stakes environment, sharpening threat recognition and response skills that could prove genuinely useful during waking life. Supporting evidence includes studies showing that children living in more dangerous or unstable environments report a higher proportion of threatening content in their dreams compared to children from safer environments, and that dream content across cultures and historical periods disproportionately features negative or threatening scenarios rather than neutral or pleasant ones, a statistical pattern that would make little sense under Freud's wish fulfillment framework but fits comfortably within a threat rehearsal explanation. A competing theory, called the continuity hypothesis, argues instead that dreams largely reflect ordinary waking concerns and experiences, rather than serving any specific evolved function at all, essentially proposing that dreaming is simply what the brain's normal information processing looks like when unconstrained by external sensory input and conscious executive control. Researchers testing this hypothesis have found that dream content does correlate meaningfully with a dreamer's recent waking experiences and preoccupations, but critics note the correlation is far weaker and more indirect than the continuity hypothesis would predict if dreams were simply straightforward replays of waking life, leaving the theory as a genuine partial explanation rather than a complete one. The specific neuroscience of what happens in the brain during dreaming has become considerably clearer since the development of functional MRI scanning, even as the deeper purpose remains debated. Dreaming activates the brain's limbic system, associated with emotional processing, considerably more strongly than it activates the prefrontal cortex, associated with logical reasoning and impulse control, a specific pattern of activation that neuroscientists studying it believe helps explain why dreams so often feel emotionally intense while simultaneously containing narrative logic that would seem absurd to the same person. While fully awake, the dreaming brain apparently generating vivid emotional content without the usual rational oversight that would flag it as nonsensical. Lucid dreaming, a state in which the dreamer becomes aware they are dreaming, while the dream is still in progress, and can sometimes exert deliberate control over its content, was long dismissed by mainstream sleep science as an unverifiable subjective claim. until a 1975 experiment conducted by British parapsychologist Keith Hearne and separately confirmed by American psychophysiologist Stephen LaBerge in 1980 established objective proof of the phenomenon. Both researchers had lucid dreamers signal the exact moment they became lucid using prearranged eye movement patterns, which, unlike the rest of the body, remain unparalyzed and physically trackable during REM sleep, and the signals arrived precisely when predicted, confirming lucid dreaming as a genuine, measurable state rather than a retrospective misremembering. Despite this confirmation, the specific neural mechanism that allows a small minority of people to achieve this unusual blend of dream state immersion and waking level self-awareness remains only partially mapped, with a 2014 German study using EEG monitoring finding increased activity in the frontal cortex during lucid episodes, a region normally quieted during ordinary dreaming, though why some people can reliably trigger this blended state and most cannot remains an open question in sleep research. A genuinely puzzling and still unresolved observation involves dream recall itself. Most people forget the overwhelming majority of their dreams within minutes of waking, typically retaining only fragments, even from dreams, recalled as vivid immediately upon opening their eyes, and researchers have proposed several competing explanations without settling on a single confirmed mechanism. One leading hypothesis points to the sharp drop in the neurotransmitter norepinephrine during REM sleep, a chemical strongly associated with memory consolidation while awake, suggesting the sleeping brain may simply lack the specific chemical conditions needed to reliably transfer dream content into long-term, retrievable memory, though this doesn't fully explain why some individuals reliably recall dreams in vivid detail across their entire lives, while others report almost never remembering a single one. Nightmares, dreams intense and distressing enough to wake the sleeper, occur disproportionately during REM sleep's later cycles, typically in the final third of a night's sleep, when REM periods grow progressively longer, and researchers studying recurring nightmare disorder, a diagnosable condition distinct from occasional bad dreams, have found a specific and genuinely useful treatment approach called imagery rehearsal therapy, first formally developed and published by psychologists Barry Krakow in 1995, in which patients consciously rewrite a recurring nightmare's ending while awake and mentally rehearse the new version repeatedly, a technique that has shown measurable success in clinical trials at reducing nightmare frequency, even though the precise mechanism by which consciously rehearsing an alternate waking narrative reshapes an involuntary sleeping one remains only partially understood at the neurological level. Further evidence that dreaming isn't unique to humans comes from a striking 2001 study conducted at MIT by neuroscientists Matthew Wilson and Kenway Louie, who recorded the firing patterns of neurons in rats' hippocampi, the brain region central to spatial memory, while the rats ran a specific maze, then continued recording as the same rats slept afterward. The researchers found the rats' neurons fired during REM sleep in patterns so closely matching the earlier maze running, sequence that the team could determine with genuine confidence which specific point in the maze the sleeping rat was effectively replaying at any given moment, some of the strongest available evidence that non-human animals experience something functionally comparable to dreaming about recent waking experience, even though no researcher can directly confirm whether that neural replay is accompanied by any subjective dreamlike experience at all. A related and still only partially explained phenomenon called REM rebound occurs when a person is selectively deprived of REM sleep specifically, while still being allowed adequate total sleep time, typically achieved in laboratory settings by waking a subject each time monitoring equipment detects the onset of REM. Once normal, uninterrupted sleep resumes, the deprived person reliably spends a significantly higher proportion of the following nights in REM sleep than they normally would, a compensatory overshoot first documented in sleep laboratory research conducted during the 1960s, strongly suggesting the brain treats REM sleep specifically rather than sleep in general, as carrying some kind of physiological necessity worth actively making up for, even though researchers still cannot say with confidence exactly what that specific necessity is. A rare and genuinely striking genetic variation discovered by neuroscientist Yinghui Fu's research team at the University of California, San Francisco, and published in 2009, involves a small number of people carrying a specific mutation in a gene called DC2, who function entirely normally on just 4 to 6 hours of sleep per night, without the accumulated cognitive impairment that severe sleep restriction reliably produces in nearly everyone else. Fu's team confirmed the effect directly by engineering mice with the identical human mutation and finding the modified mice also slept substantially less than normal, while showing no measurable decline in performance on standard rodent cognitive tests, genuine evidence the mutation itself, rather than some other lifestyle factor, was responsible. These naturally short sleepers remain exceedingly rare, and researchers have since identified a small handful of additional genetic variants associated with the same trait, but the specific biological mechanism explaining why disrupting normal sleep duration proves so reliably harmful to almost everyone else. While these particular individuals experience no apparent cost at all, remains only partially understood. Sleep paralysis, a distinct and genuinely unsettling phenomenon, experienced by an estimated 8% of the general population at least once, according to a large 2011 meta-analysis of more than 36,000 subjects, occurs when a person becomes consciously aware, while the REM atonia discussed earlier in tonight's video is still physically active, leaving them fully awake and alert, yet completely unable to move or speak, an experience frequently accompanied by vivid, often frightening hallucinations of a threatening presence in the room. Researchers studying the phenomenon believe it results from a specific misalignment in the normal transition between REM sleep and full wakefulness, and cross-cultural anthropological research has found strikingly similar folkloric explanations for the same physiological experience across entirely unconnected societies, including the Newfoundland Old Hag, the Japanese Kanashibari, and much of Western Europe's historical incubus and succubus mythology, all independently, describing an oppressive, immobilizing nighttime presence, evidence that a shared underlying neurological experience likely produced strikingly similar folk explanations across cultures with no direct contact, even though the specific neural trigger that occasionally causes the REM atonia and waking consciousness systems to briefly overlap remains only partially mapped. A related and genuinely stranger hypnagogic phenomenon called exploding head syndrome, despite involving no actual pain or physical harm, causes the affected person to perceive a sudden, extremely loud noise, often described as a gunshot, explosion, or crashing cymbal occurring at the exact moment of falling asleep, or briefly waking with no external sound actually present. First formally named and described in a 1988 medical paper by neurologist J.M.S. Pierce, the condition is now believed to affect a meaningful minority of the population, at least occasionally, and the leading, though still unconfirmed, explanation proposes it results from a kind of neural misfire during the transition into sleep, in which the brainstem's auditory processing regions briefly and erroneously activate all at once, in a burst resembling how those same regions would normally respond to a genuinely loud external sound. Though why this particular sensory system misfires in this specific auditory way, rather than through some other sense during the sleep transition, remains unexplained. Running the length of your digestive tract, from esophagus to rectum, sits a network of more than 500 million neurons called the enteric nervous system, a number roughly equivalent to the entire nervous system of a cat, and this network is capable of operating your digestion almost entirely independently of your brain and spinal cord. A discovery substantial enough that many gastroenterologists and neuroscientists have taken to calling it, informally but seriously, the body's second brain. The enteric nervous system was first seriously investigated in detail by American neuroscientist Michael Gershon, whose 1998 book, The Second Brain, brought the concept to widespread public and scientific attention, building on research showing that if the vagus nerve, the primary connection between gut and brain, is completely severed, the digestive tract continues to coordinate the complex muscular contractions of digestion, called peristalsis, entirely on its own, using its own local reflex circuits and its own supply of more than 30 neurotransmitters, including roughly 95% of the body's total serotonin, a chemical more commonly associated with mood regulation in the brain, produced instead primarily within the gut. That last fact, the sheer concentration of serotonin manufactured in the digestive system rather than the brain, has become the foundation for an entire and still rapidly developing field of research called the gut-brain axis, studying the bidirectional communication between digestive function and mental state. Researchers have found that the trillions of bacteria living within the gut microbiome appear capable of influencing this communication directly, and a landmark 2011 study conducted at McMaster University in Canada found that mice raised in completely sterile, germ-free environments, lacking any gut bacteria at all, displayed measurably different anxiety-related behaviors compared to normally raised mice, and that transplanting gut bacteria from one strain of mice known for bold behavior into germ-free mice of a more anxious strain measurably shifted the recipient mice's behavior toward boldness, a genuinely striking result suggesting gut bacteria can influence behavior through some pathway scientists are still working to fully map. The human gut microbiome itself contains an estimated 38 trillion bacterial cells, a number establishing, according to a widely cited 2016 recalculation published by researchers Ron Sender, Shai Fuchs, and Ron Milo, that bacterial cells in the body roughly equal the total number of human cells, overturning an older and less precise estimate that had bacteria outnumbering human cells by a factor of 10. Each person's specific microbiome composition is almost as individually distinctive as a fingerprint, shaped by factors including birth method, since babies delivered vaginally acquire a substantially different initial bacterial population than those delivered by cesarean section, a difference some researchers have linked, though not yet conclusively, to later differences in immune system development and allergy risk. A particularly striking and still only partially explained phenomenon involves the gut's direct influence on food cravings and eating behavior. Research published through the 2010s has found that certain gut bacteria appear to produce specific signaling molecules that influence appetite and food preference, effectively creating a feedback loop in which particular bacterial populations may increase cravings for the specific foods that best allow those same bacteria to thrive. Most notably, sugar-metabolizing species favoring sugar-rich diets, a relationship researchers studying it have described as the gut microbiome potentially exerting a genuine, measurable influence over conscious food choices, though the exact biochemical signaling pathways involved remain an active area of ongoing research rather than a fully solved mechanism. The vagus nerve itself, the primary physical highway connecting gut and brain, carries a genuinely surprising traffic pattern. Approximately 80 to 90% of its nerve fibers carry information upward from gut to brain, rather than downward from brain to gut, meaning the digestive system sends the brain far more information than it receives back, a ratio that has led some researchers studying the gut-brain axis to argue the traditional assumption that the brain is always the body's primary command center deserves genuine reconsideration, at least with respect to how much raw sensory, chemical information actually flows in each direction along this particular connection. Irritable bowel syndrome, a chronic condition affecting an estimated 10 to 15% of people worldwide. According to global epidemiological surveys conducted through the 2010s, remains a genuinely instructive example of how much the gut-brain relationship still isn't fully understood even in a clinically significant, widely studied condition. Patients with IBS show no detectable structural damage or disease markers in standard diagnostic testing, and researchers have proposed several contributing mechanisms, including altered gut microbiome composition, heightened visceral pain sensitivity, and dysregulated communication along the gut-brain axis itself, without any single confirmed cause, leaving IBS formally classified as a functional disorder. Medical terminology essentially acknowledging that the symptoms are real and measurable, while the precise underlying mechanism generating them remains incompletely explained. The appendix, a small pouch attached to the large intestine, long dismissed as a purely vestigial organ with no remaining function, has undergone a genuine scientific reconsideration over the past 2 decades. A 2007 paper published by immunologist William Parker and colleagues at Duke University proposed the appendix functions as a protected reservoir for beneficial gut bacteria. Its narrow, tucked-away structure sheltering a small reserve population from illnesses like severe diarrhea that can otherwise wipe out much of the gut's bacterial population, allowing the appendix to help reseed and restore a healthy microbiome afterward. Supporting evidence gathered since includes a large 2011 epidemiological study finding that people who had undergone appendix removal were more likely to experience a recurrence of a specific dangerous intestinal infection called Clostridioides difficile, consistent with the reservoir hypothesis, though the appendix's precise overall contribution to long-term health remains difficult to measure directly, since the vast majority of people who have it removed go on to live entirely normal lives with no detectable difference in gut health. A separate and only recently formalized area of research involves interoception, the body's capacity to sense its own internal physiological state, heartbeat, hunger, breathing rate, and the general hard-to-name feeling of a gut instinct. Neuroscientist Sarah Garfinkel and colleagues, publishing research through the 2010s, found measurable individual differences in interoceptive accuracy, tested most commonly by asking subjects to silently count their own heartbeats without touching their pulse, and found people with higher measured interoceptive accuracy also reported more intense emotional experiences generally, suggesting the physical sensing of one's own internal bodily state may be more tightly bound to conscious emotion than previously assumed, a connection that remains an active area of psychiatric research, given documented links between disrupted interoception and conditions including anxiety disorders and anorexia nervosa, though the precise causal direction, whether poor interoception contributes to these conditions or results from them, remains unresolved. The clinical power of the gut microbiome received one of its most dramatic real-world demonstrations through fecal microbiota transplantation, a treatment in which stool from a healthy donor containing that donor's full bacterial population is transferred into the intestine of a patient suffering from a severe recurrent infection of Clostridioides difficile, a bacterium that can overwhelm the gut once its normal bacterial population has been depleted, often by extended antibiotic use. A landmark randomized clinical trial published in 2013 in the New England Journal of Medicine, led by physician Els van Nood, found the procedure cured recurrent C. difficile infection in 94% of patients after 1 or 2 transplants, dramatically outperforming standard antibiotic treatment, a result striking enough that the trial was stopped early on ethical grounds once the treatment's superiority became statistically undeniable. Despite the treatment's now well-established clinical success and its formal approval for this specific use, researchers still cannot fully explain precisely which components of a healthy donor's bacterial population are doing the essential curative work, since transplants using only specific isolated bacterial strains rather than a donor's complete, complex microbial community have so far produced less consistent results, suggesting the treatment's effectiveness may depend on a genuinely complex ecological restoration rather than any single identifiable beneficial species. A further, genuinely contested area of gut research involves intestinal permeability, informally known as leaky gut, referring to the idea that the tight junctions between cells lining the intestinal wall, which normally regulate exactly what passes from the gut into the bloodstream, can become abnormally loose, allowing bacterial fragments and partially digested food particles to leak through and trigger low-grade immune activation elsewhere in the body. Researcher Alessio Fasano's work identifying a specific protein called zonulin, published beginning in 2000, provided a genuine molecular mechanism showing intestinal permeability could be actively regulated rather than simply fixed, and increased permeability has since been confirmed in specific, well-documented conditions, including celiac disease. Where the science remains genuinely unsettled is the considerably broader popular claim that leaky gut independently causes a wide range of unrelated conditions, from chronic fatigue to autoimmune disease to mood disorders, a claim mainstream gastroenterology organizations have generally treated with caution, since rigorous controlled evidence directly linking measured intestinal permeability to most of these broader claimed conditions remains limited, leaving zonulin and intestinal permeability a genuine and actively studied biological mechanism whose full clinical significance is still being carefully worked out rather than a settled explanation for the wide range of ailments sometimes attributed to it. The gut microbiome also functions as a genuine, ongoing biochemical factory, and among its more clinically significant outputs are specific vitamins. The human body cannot manufacture on its own, most notably vitamin K2, essential for proper blood clotting, and vitamin B12, essential for nerve function and red blood cell production, both synthesized in meaningful quantities by resident gut bacterial species. Researchers studying newborn infants, whose gut microbiome hasn't yet had time to establish a full bacterial population, have found this specific vulnerability directly reflected in a real and well-documented medical condition, vitamin K deficiency bleeding, a risk serious enough that a preventive vitamin K injection is now standard practice at birth in most developed countries, specifically to cover the gap before an infant's own gut bacteria mature enough to reliably produce sufficient quantities on their own. Precisely how much of an adult's total vitamin K2 and B12 needs are met through bacterial production alone, as opposed to dietary intake, varies considerably between individuals depending on their specific microbiome composition, a variability that remains only partially mapped, leaving nutritional science with a genuine open question about how much of what researchers currently classify as an individual's baseline dietary requirement is actually a joint product of diet and a person's own particular bacterial population working together. French military surgeon Ambroise Paré provided one of the earliest documented medical descriptions of phantom limb sensation in 1552, recording that soldiers who had undergone amputation frequently reported feeling pain in the limb that no longer physically existed, and more than 450 years later, despite phantom limb sensation now being confirmed in an estimated 60 to 80% of amputees, according to modern clinical surveys, the precise mechanism generating the sensation remains only partially resolved. The most influential modern explanation, developed primarily by neuroscientist V.S. Ramachandran beginning in the early 1990s, centers on the brain's somatosensory cortex, the strip of neural tissue that maintains a detailed internal map of the entire body's surface. Ramachandran proposed that when a limb is amputated, The brain region that once processed sensation from that limb doesn't simply go silent, but instead gets gradually invaded by neural signals from neighboring body map regions, a process called cortical remapping, so that touching a different part of the body, in some well-documented cases the face, can trigger a sensation the amputee perceives as originating from the missing limb entirely. Ramachandran's own famous 1990s experiments used a simple mirror box, reflecting the amputee's intact limb to visually replace the missing one, and found that many patients experienced genuine, if temporary, relief from phantom pain simply by watching the mirrored reflection perform movements, evidence that the brain's visual and body mapping systems are far more intertwined and far more capable of being deliberately tricked than earlier pain models had assumed. Phantom limb sensation isn't limited to amputees. A genuinely striking related phenomenon occurs in a small percentage of people born without a limb at all, who nonetheless sometimes report a phantom sensation for a body part they never physically possessed, a finding that has pushed researchers toward concluding at least part of the body's internal map may be substantially pre-wired into the brain from birth, rather than built up entirely through lived physical experience. Though the precise balance between innate and experience-shaped body mapping remains an actively studied and unresolved question, pain itself, even setting aside the specific phantom limb puzzle, remains a genuinely strange biological signal in ways most people never have reason to consider. The specific intensity of pain a person experiences from an identical physical injury varies enormously between individuals, and researchers studying this variation have identified a specific genetic factor, a particular variant of a gene called SCN9A, first characterized in a landmark 2006 study of Pakistani families, in which several children were found to feel no physical pain at all, despite otherwise normal nerve function, allowing them to suffer repeated serious injuries, including bone fractures and burns, without ever registering pain as a warning signal. Studying these rare, pain-insensitive individuals directly led pharmaceutical researchers toward SCN9A as a specific and promising target for new non-opioid pain medications, since blocking the exact channel this gene controls could, in principle, replicate the same pain-blocking effect these families experience naturally, without the broader central nervous system risks associated with opioid painkillers, research that remains in active clinical development as of the most recent published trial updates. An entirely different and genuinely counterintuitive pain-related condition called congenital insensitivity to pain with anhidrosis involves not just an absence of pain sensation, but also an inability to sweat, and researchers, studying the small number of documented cases, have found the condition results from mutations affecting a different gene, NTRK1, which is required for the proper development of the specific nerve fibers responsible for both pain sensation and sweat gland activation, a genuinely unexpected biological link between the ability to feel a burn and the ability to physically cool down from one. Placebo analgesia, the measurable reduction in pain some patients experience after receiving a treatment with no active pharmacological ingredient at all, offers a further genuine puzzle within pain research, precisely because brain imaging studies conducted since the early 2000s have confirmed the effect is not merely psychological in the dismissive, it's all-in-your-head sense, but involves the brain's own genuine production of endogenous opioids, the body's natural pain-relieving chemicals, in response to the expectation of relief alone. A landmark 1978 study by researchers John Levine, Newton Gordon, and Howard Wolves found that administering naloxone, a drug that specifically blocks opioid receptors, substantially reduced the placebo effect in patients recovering from dental surgery. Direct evidence that the brain was generating real, measurable opioid activity. purely from belief and expectation, a finding replicated many times since, and one that has meaningfully reshaped how seriously medical researchers treat the placebo response, even as the precise psychological and neurological triggers that determine why placebo analgesia works powerfully in some patients and barely at all in others remain incompletely mapped. The basic question of how a physical sensation like a pinprick becomes registered by the brain as painful at all was substantially reframed by the gate control theory of pain, proposed by psychologist Ronald Melzack and physiologist Patrick Wall in a landmark 1965 paper published in the journal Science. Before gate control theory, pain was generally treated as a simple direct signal traveling from injury to brain along a fixed pathway, but Melzack and Wall proposed instead that a specific gate mechanism within the spinal cord actively regulates how much pain signal actually reaches the brain, capable of being partially closed by other simultaneous sensory input, an insight that helped explain the previously puzzling, everyday observation that rubbing or shaking an injured area genuinely does reduce perceived pain, since the added touch sensation competes for the same neural gateway. Gate control theory has since been substantially refined at the molecular level, but its core insight that pain is actively modulated by the nervous system rather than simply relayed remains foundational to modern pain medicine and continues to inform treatments like transcutaneous electrical nerve stimulation, which deliberately floods the same spinal gateway with a competing non-painful electrical signal. Itch, a sensation closely related to pain but processed through a genuinely distinct set of nerve fibers, remained considerably less scientifically understood for decades longer, and the specific molecule most responsible for chronic itch, a receptor called MRGPRA3, was only identified through mouse studies published in 2009. What makes itch a particularly strange sensation is its well-documented contagious quality, since simply watching another person scratch reliably triggers a measurable urge to scratch in an observer, an effect confirmed through controlled studies, including a 2012 experiment led by dermatologist Gil Josipovich and brain imaging conducted during induced itching sessions, has found activation patterns overlapping considerably with brain regions associated with the urge to move and with empathic response to others, though why itch specifically, more so than most other bodily sensations, spreads so reliably just from visual observation remains only partially explained. Chronic pain, pain that persists for months or years after any original injury has fully healed, has led researchers to a genuinely significant reframing of pain itself, centered on a phenomenon called central sensitization, in which the spinal cord and brain's own pain processing circuits become progressively more sensitive over time, eventually generating a strong pain signal stimuli that wouldn't normally register as painful at all, or continuing to generate pain long after the original tissue damage has resolved. Neuroscientist Clifford Wolf's foundational 1983 research first demonstrated this kind of central nervous system sensitization directly in animal studies, showing that repeated painful stimulation could leave the spinal cord itself measurably more reactive to subsequent, otherwise mild stimuli, evidence that chronic pain in at least some patients isn't simply an ongoing signal from damaged tissue, but a genuine, separate change in how the nervous system itself processes sensation, a distinction with significant treatment implications, since it suggests chronic pain sometimes needs to be treated as a nervous system disorder in its own right rather than solely as a symptom pointing back toward an original, ongoing physical injury, though predicting in advance which patients will develop this kind of lasting sensitization after an injury, and which will not remains an unresolved clinical challenge. Referred pain, in which an injury or disease affecting one internal organ is perceived by the brain as originating from an entirely different, often distant part of the body, presents a further genuinely counterintuitive puzzle within pain research, the clearest and clinically most significant example being a heart attack, which classically produces pain radiating down the left arm or into the jaw, rather than being clearly localized to the chest alone. The leading explanation, called the convergence projection theory, first proposed in the 1890s and refined considerably through 20th century neuroscience, holds that sensory nerve fibers from internal organs and from the corresponding referred skin area converge onto the very same neurons within the spinal cord before continuing up to the brain. And because the brain has, over a lifetime, received vastly more sensory information from the skin than from internal organs, it defaults to interpreting the combined signal as coming from the more familiar external location. The theory accounts reasonably well for the general pattern of referred pain, but doesn't fully explain why the specific referral pattern varies somewhat between individuals experiencing the identical underlying medical event, a variability that has led some emergency physicians to note with genuine clinical concern that atypical or absent referred pain, more common in women and older patients experiencing heart attacks, according to multiple published studies, can contribute to delayed diagnosis precisely because the expected textbook referral pattern doesn't always appear. Charles Darwin devoted an entire chapter of his 1872 book, The Expression of the Emotions in Man and Animals, to blushing, calling it, in his own words, the most peculiar and the most human of all expressions. And more than 150 years later, the specific evolutionary purpose of blushing remains one of the more genuinely debated open questions in the study of human emotional expression. Blushing occurs when the sympathetic nervous system, triggered by embarrassment, social scrutiny, or self-consciousness, causes blood vessels in the face to dilate, a physical response that, unlike most emotional reactions, cannot be consciously suppressed or faked, a fact confirmed through numerous physiological studies, and one that makes blushing genuinely unusual among human emotional displays. The leading evolutionary explanation, developed and refined by psychologist Dr. Keltner and colleagues through research published beginning in the mid-1990s, proposes that blushing functions as an involuntary, unfakeable signal of social remorse or awareness of having violated a social norm, and that observers unconsciously trust and forgive a blushing person more readily than a non-blushing one, precisely because the reaction cannot be deliberately performed, a theory supported by several controlled experiments in which participants rated identical social transgressions more sympathetically when the transgressor was shown blushing afterward. What remains less settled is why humans specifically evolved this particular unfakeable signal rather than some other equally honest one, and why no other primate species shows anything resembling the same reliable, involuntary facial blood vessel response to social embarrassment. Goosebumps, formerly called piloerection, present a related evolutionary puzzle involving what researchers generally agree is a genuinely vestigial reflex, a leftover from an ancestral trait that no longer serves its original function in modern humans. In mammals with thick fur, piloerection causes hair to stand erect, trapping an insulating layer of air for warmth in cold conditions, and making the animal appear physically larger when threatened, both clearly useful functions for a furred ancestor. Modern humans retain the identical muscular reflex, tiny erector pili muscles at the base of each hair follicle, contracting in response to cold or strong emotion, despite having nowhere near enough body hair for the reflex to provide any meaningful warmth or size-related benefit, making goosebumps one of the clearest and most frequently cited examples of vestigial human anatomy, a physical reflex that has outlived the biological function it originally evolved to serve. What remains genuinely less well explained is why goosebumps also reliably occur in response to certain intense emotional or aesthetic experiences entirely unrelated to cold or physical threat, most notably powerful music, a phenomenon researchers call frisson. A 2016 study conducted by researcher Matthew Sachs at the University of Southern California found that people who reliably experience musical frisson show measurably different brain structure in regions connecting the auditory cortex to areas involved in emotional processing, compared to people who rarely or never experience the sensation, suggesting a genuine underlying neurological difference between individuals rather than simple variation in musical taste, though why an ancestral cold and threat reflex became linked to aesthetic and emotional experiences that have nothing to do with either temperature or danger remains an open evolutionary question. Hiccups present a genuinely similar evolutionary puzzle. An involuntary spasm of the diaphragm, paired with a sudden closure of the vocal cords that serves no identified purpose in adult human physiology and that even causes measurable discomfort. One prominent evolutionary hypothesis, published by French researcher Christian Strauss and colleagues in 2003, proposes hiccups are a vestigial reflex inherited from an amphibian ancestor's method of pushing water across gills while keeping it out of developing lungs, a movement pattern the hypothesis suggests briefly reappears in human fetuses and infants who hiccup considerably more frequently than adults, before mostly fading as the reflex loses whatever adaptive value it once carried. The hypothesis remains genuinely contested rather than confirmed, and no single trigger reliable explains every case of hiccups in adults, leaving remedies passed down informally: holding one's breath, drinking water upside down, being startled, without any of them backed by a clearly demonstrated shared physiological mechanism. Emotional tears present perhaps the strangest entry in this entire chapter, since humans are the only species confirmed to cry tears specifically in response to emotional states rather than purely as a physical, reflexive response to irritation or injury. Biochemist William Frey conducted one of the more frequently cited studies on the subject, published in 1985, comparing the chemical composition of emotional tears against tears produced by chopping onions in the same subjects, and found emotional tears contained measurably higher concentrations of certain stress-related hormones and proteins, including adrenocorticotropic hormone, leading Frey to propose that emotional crying might function as a mechanism for physically expelling stress-related chemicals from the body. This specific tears as detox hypothesis has never been definitively confirmed as crying's actual evolutionary purpose, and other researchers have proposed competing explanations centered on tears functioning primarily as a social signal, since visible crying reliably triggers comfort and support responses in observers, a finding supported by several controlled studies, leaving the precise evolutionary reason. Humans specifically evolved emotional tears, unlike any other studied species, still without a single confirmed, universally accepted explanation. Contagious yawning presents a final, genuinely strange entry in this catalog of involuntary human signals, the well-documented tendency for simply seeing, hearing, or even reading about another person yawning to trigger a yawn of one's own, an effect confirmed in controlled studies to occur in roughly 40 to 60% of people exposed to a yawning stimulus persons. Neuroscientists studying the phenomenon have found it correlates with measures of empathy, with a 2010 study led by researcher Elizabeth Cerulli finding that people who scored lower on standard empathy assessments were also significantly less susceptible to contagious yawning, and functional imaging studies have implicated the same mirror neuron system thought to be involved in imitating others' movements. More generally, though the specific evolutionary function of contagious yawning itself remains genuinely debated, with competing hypotheses proposing it may help synchronize alertness levels across a social group, a behavior also documented in chimpanzees and, according to a 2008 study, even in domestic dogs watching a human yawn. Tears carry a further layer of biological function well beyond the emotional signaling discussed above, a discovery that traces back somewhat unexpectedly to bacteriologist Alexander Fleming, considerably better known for his later 1928 discovery of penicillin. In 1922, Fleming noticed that a drop of his own nasal mucus, added to a bacterial culture he was growing, caused the surrounding bacteria to dissolve, and following up on the observation, he found the same dissolving effect occurred with tears, identifying an enzyme. He named lysozyme, present in tears, saliva, and mucus, capable of breaking down the cell walls of many types of bacteria on contact. Lysozyme's antibacterial function has since been confirmed and studied in considerably greater molecular detail, and researchers have found tear composition changes measurably in response to different triggers, with emotional tears, reflex tears from irritation, and the continuous basal tears that keep the eye lubricated, all differing slightly in their specific protein content, though scientists still don't have a complete account of exactly how the lacrimal gland adjusts its output so precisely to match three such different biological triggers. Laughter, along with the closely related sensation of being tickled, rounds out this chapter's catalog of involuntary human responses with a genuinely peculiar built-in exception. Nearly everyone finds it difficult or entirely impossible to tickle themselves, a phenomenon formally investigated through brain imaging research, published in 1998 by neuroscientist Sarah Jane Blakemore and colleagues at University College London. Blakemore's team found that the cerebellum, a brain region responsible for predicting the sensory consequences of one's own movements, generates an advanced signal that effectively cancels out the anticipated sensation whenever a touch results directly from one's own hand movement, a mechanism that ordinarily serves the useful purpose of letting the brain distinguish self-generated sensations from external ones, but that, as a direct side effect, dampens the ticklish response specifically because a self-inflicted tickle is entirely predictable to the cerebellum in a way an external tickle from someone else never is. The finding helps explain the self-tickling exception mechanically, but the deeper evolutionary question of why ticklishness and its accompanying laughter evolved as a response to specific, predictable areas of unexpected external touch in the first place remains genuinely debated, with competing hypotheses proposing it may have originated as a playful mechanism for practicing defensive reflexes in vulnerable body areas during juvenile social play. A rare neurological condition called pseudobulbar affect offers a genuinely unsettling illustration of how disconnected the outward expression of laughing or crying can become from the actual emotion supposedly driving it. The condition, which can result from a range of underlying neurological damage, including stroke, traumatic brain injury, and certain neurodegenerative diseases, causes sudden, uncontrollable episodes of laughing or crying that are disproportionate to, or entirely disconnected from, the person's actual felt emotional state at the time, with patients frequently reporting they feel only mildly amused or not upset at all during an episode of intense, uncontrollable laughter or tears. Researchers studying the condition, formally characterized in detail through neurological case studies accumulating since the late 20th century, have traced it to damage in specific neural circuits connecting the cerebellum, brainstem, and frontal cortex, circuits normally responsible for smoothly regulating and appropriately scaling the outward physical expression of emotion to match its actual felt intensity. And the condition's existence has become genuinely useful evidence for neuroscientists that the felt experience of an emotion and its outward physical expression are produced by at least partially separate neural systems that can, under the right kind of damage, become measurably decoupled from one another. The human immune system operates as two genuinely distinct defense networks working in close coordination: the innate immune system, which responds rapidly but generically to any perceived threat, and the adaptive immune system, which develops highly specific, targeted responses to particular pathogens and, remarkably, retains a functional memory of them, sometimes for the entire remainder of a person's life. This immunological memory represents one of biology's genuinely more elegant unsolved puzzles, since a specific class of white blood cells, called memory B cells and memory T cells, first identified in detailed form through immunology, Research conducted across the 1960s and refined considerably since can persist in the body for decades after an initial infection has fully cleared, ready to mount a dramatically faster and stronger response if the same pathogen ever returns. Landmark study published in 2003 by immunologist Rafi Ahmed's research team at Emory University found measurable levels of antibody-producing memory cells against the 1918 influenza virus still present and functional in elderly survivors of that pandemic more than 80 years after their original infection. Direct evidence that at least some forms of immune memory can persist for essentially an entire human lifetime, even though the precise cellular mechanisms that keep these memory cells alive and functional for so many decades without simply dying off like most cells in the body remain only partially understood at the molecular level. Vaccination works by deliberately triggering this same memory-forming process without requiring an actual dangerous infection first, and the specific immunological principle behind it was first demonstrated without any understanding of the underlying cellular mechanism by English physician Edward Jenner in 1796, when he inoculated a boy named James Phipps with cowpox material, and later showed the boy had become protected against the considerably more dangerous smallpox virus, nearly 2 full centuries before scientists could actually explain, at the cellular and molecular level, precisely why exposure to one virus could confer protection against a related but distinct one. A separate and genuinely puzzling immunological phenomenon involves the placenta, the temporary organ that allows a developing fetus, which carries a genetically distinct set of proteins inherited half from the father, to grow inside a mother's body for 9 months without the mother's own immune system attacking it as foreign tissue, the way it would almost certainly attack a comparably mismatched organ transplant. Researchers have identified several contributing mechanisms, including specialized immune-suppressing cells concentrated specifically at the maternal-fetal boundary and unique molecular signals produced by placental tissue that appear to actively instruct nearby immune cells to stand down. But immunologists studying pregnancy still describe the full picture of fetal immune tolerance as incompletely mapped, an active area of ongoing research with direct clinical relevance to conditions like preeclampsia, in which this normal immune tolerance appears to partially break down. The human microbiome's relationship with the immune system extends well beyond the gut-brain axis discussed earlier in tonight's video, since a majority of the body's total immune cells, by some published estimates as much as 70 to 80%, are located directly within or immediately surrounding the gut lining, in constant, close contact with the trillions of resident gut bacteria. Researchers studying this relationship have found that the immune system doesn't simply tolerate this bacterial population passively, but appears to actively distinguish beneficial resident bacteria from genuinely dangerous pathogens using a specific set of receptor proteins called pattern recognition receptors, first characterized in detail through research published by immunologists Jules Hoffman and Bruce Beutler, work that earned them a share of the 2011 Nobel Prize in Physiology or Medicine, though precisely how the immune system reliably learns to tolerate an entirely new individual's specific bacterial population during infancy, rather than attacking it as foreign, remains only partially resolved. Autoimmune disease, in which the immune system's normal ability to distinguish the body's own tissue from foreign threats breaks down and begins attacking healthy cells directly, affects an estimated 24 million people in the United States alone, according to National Institutes of Health. Figures spread across more than 80 distinct recognized conditions including rheumatoid arthritis, lupus, and multiple sclerosis, and despite decades of dedicated research, no single unifying cause has ever been identified across this entire disease category. Researchers have identified contributing genetic risk factors for several specific autoimmune conditions and have proposed a leading general hypothesis called molecular mimicry, in which a bacterial or viral protein happens to closely resemble a normal human protein, closely enough that antibodies trained to attack the pathogen mistakenly begin attacking the similar-looking human tissue as well, a mechanism confirmed in a small number of specific, well-documented cases. Though why any individual immune system develops this specific self-targeting error, and why autoimmune conditions disproportionately affect women at roughly a 4-to-1 ratio compared to men, according to consistent epidemiological findings across multiple large studies, remain among the field's most significant open questions. Fever, the deliberate elevation of body temperature the immune system triggers during many infections, has long been understood at a basic mechanical level, driven by signaling molecules called pyrogens acting on the brain's hypothalamus. Yet its precise adaptive value remains a subject of genuine ongoing research rather than settled fact. The leading explanation holds that a raised body temperature directly impairs the reproduction of many bacteria and viruses, while simultaneously enhancing the activity of several immune cell types, a hypothesis supported by studies including a widely cited 1975 experiment on desert iguanas, cold-blooded animals that, when infected, deliberately sought out warmer environments to raise their own body temperature, and researchers who prevented the iguanas from doing so found a significantly higher mortality rate from infection, suggesting fever's benefit was substantial enough to evolve independently as a deliberate behavioral strategy, even in animals that cannot generate the heat internally. A related and still genuinely contested explanation for the sharp rise in allergic and autoimmune conditions across industrialized nations over the past century is the hygiene hypothesis, first proposed by epidemiologist David Strachan in a 1989 paper after he found that children from larger families who statistically experience more childhood infections from siblings showed measurably lower rates of hay fever and eczema later in life. The hypothesis proposes that reduced early life exposure to a broad range of microbes, a consequence of modern sanitation, antibiotics, and smaller family sizes, may leave the developing immune system without enough varied stimulation, causing it to become more prone to misdirecting its response toward harmless substances like pollen, or in autoimmune cases, the body's own tissue. The hygiene hypothesis has been partially revised since Strachan's original formulation, with many immunologists now favoring a more specific version focused on exposure to beneficial environmental microbes and gut bacteria specifically rather than infections generally. But the broader question of exactly how much of the modern rise in allergic and autoimmune disease can be attributed to changed microbial exposure as opposed to other genetic or environmental factors remains an active and unresolved area of research. For most of modern immunology's history, only the adaptive immune system, the branch responsible for the antibody-based memory discussed earlier in this chapter, was believed capable of remembering a prior threat. That assumption was directly challenged by research into a phenomenon called trained immunity, formally proposed and named in an influential 2011 paper by immunologist Mihai Netea, which found that the innate immune system, long assumed to respond identically to any threat regardless of prior exposure, can also retain a kind of non-specific memory, becoming measurably more responsive to a wide range of subsequent infections after certain earlier exposures. The clearest evidence comes from long-term studies of the BCG vaccine, originally developed against tuberculosis, which multiple epidemiological studies conducted primarily in West Africa found was associated with significantly reduced overall childhood mortality from causes entirely unrelated to tuberculosis, a broader protective effect. Researchers eventually traced to epigenetic changes in innate immune cells themselves, essentially reprogramming those cells to respond more aggressively to a wide range of subsequent threats rather than training them to recognize one specific pathogen a genuinely significant revision to a decades-old assumption about how immune memory works, though exactly how long trained immunity's protective effects last and precisely which other vaccines or exposures might trigger a similar broad benefit remains under active investigation. A large twin study published in 2015 by immunologist Mark Davis and colleagues at Stanford University produced a genuinely significant and somewhat humbling reframing of how much of immune function is actually determined by genetics at all. Studying 210 twin pairs, including both identical twins, who share essentially all of their genes, and fraternal twins who share roughly half, Davis's team measured more than 200 separate immune system parameters and found that the large majority, roughly 3/4, were shaped considerably more by environmental factors, prior infections, diet, and other lived experience than by genetics, with identical twins showing measurably divergent immune profiles despite their genetic identity, a divergence that grew more pronounced with age as the twins accumulated increasingly different life exposures. The finding meaningfully complicated an older assumption that immune function was primarily hardwired at birth and helped explain why identical twins, despite genetically identical starting points, can nonetheless show significantly different susceptibility to infections and different responses to the same vaccine later in life, though researchers still cannot fully predict for any given individual exactly which specific environmental exposures will shape their immune profile in which particular direction. A small number of specific tissues in the body, most notably the eye's interior and the brain, occupy what immunologists call a state of immune privilege, meaning the ordinary, aggressive immune response mounted almost everywhere else in the body is deliberately dialed back within these specific regions, a concept first identified experimentally in the 1940s by researcher Peter Medawar, who found that tissue grafts placed inside the eye's anterior chamber survived considerably longer without rejection than identical grafts placed elsewhere in the body. The evolutionary logic is generally understood well enough. Both the eye and the brain contain delicate, largely non-regenerating tissue where a normal, inflammation-heavy immune response, however effective at clearing infection elsewhere, would risk causing more permanent functional damage than the infection itself. And researchers have identified several specific contributing mechanisms, including the blood-brain barrier's tight physical restriction on which immune cells can enter brain tissue at all, and specific local signaling molecules that actively suppress immune activity within the eye. What remains only partially resolved is how this privileged status is maintained without leaving these tissues catastrophically vulnerable to infection, since certain pathogens, including the herpes simplex virus, responsible for some forms of eye infection, appear specifically adapted to exploit this reduced immune surveillance, meaning the same evolved protection that shields these delicate tissues from immune damage, can also, in specific documented cases, provide a genuine foothold for exactly the kind of infection it was meant to help guard against. When the Human Genome Project completed its initial full sequencing of human DNA in 2003, after a 13-year international research effort, one of its most genuinely surprising findings was just how little of the genome actually codes for proteins, the specific molecules that carry out most of the body's cellular functions. Protein coding sequences make up only an estimated 1 to 2% of the entire human genome, leaving the remaining roughly 98% originally dismissed. In a term coined by geneticist Susumu Ono in 1972 as junk DNA, a label that has since become one of the more actively contested in modern genetics. The Encyclopedia of DNA Elements Project, commonly abbreviated ENCODE, a large international research consortium that published its first major findings in 2012 after nearly a decade of coordinated research, reported that a substantial majority of the genome, by the consortium's own published estimate at least 80%, shows some form of measurable biochemical activity, including regulatory functions that influence when and how nearby genes are switched on or off, rather than sitting entirely inert as the earlier junk label implied. This finding generated genuine and still ongoing scientific controversy, since a considerable number of geneticists, including a widely cited critical 2013 paper by researchers Dan Grauer and colleagues, argued the ENCODE consortium's 80% figure dramatically overstated genuine biological function by counting any detectable biochemical activity, however minor or incidental, as evidence of functional importance, a methodological disagreement that remains genuinely unresolved within the field, with most geneticists now generally agreeing the true proportion of the genome serving a confirmed meaningful biological function likely sits somewhere considerably lower than ENCODE's original 80% figure, but almost certainly higher than the roughly 2% implied by the original protein coding estimate alone. Among the specific categories of non-coding DNA that have moved most dramatically from presumed junk to confirmed function are microRNAs, tiny genetic sequences first identified in detail through research on roundworms published by Victor Ambrose and Gary Roof-Kuhn. In 1993, a discovery that earned both researchers the 2024 Nobel Prize in Physiology or Medicine. MicroRNAs don't code for proteins at all, but instead directly regulate which existing genes get expressed and to what degree, functioning as a genuinely sophisticated genetic control layer that researchers now understand plays a significant role in normal development and, when disrupted in a range of diseases including several cancers, a functional importance that would have been entirely invisible under the genome's original protein coding-only framework for what counted as meaningful genetic material. Transposable elements, informally called jumping genes, make up an even larger share of the human genome than protein coding sequences, an estimated 45% by most current published measurements, and consist of DNA sequences capable of copying and reinserting themselves at new locations. Throughout the genome, a phenomenon first discovered by geneticist Barbara McClintock through her research on maize, plants conducted primarily during the 1940s and 1950s, work that went largely unrecognized by the broader scientific community for decades before eventually earning her the unshared 1983 Nobel Prize in Physiology or Medicine. The overwhelming majority of these transposable elements have accumulated mutations over evolutionary time that have permanently disabled their ability to actually move anymore, effectively fossilized remnants of ancient genetic activity. But a small number remain genuinely active. Even in modern humans, and researchers have documented specific rare cases in which an active transposable element inserting itself into a critical gene has directly caused a genetic disease, including certain documented cases of hemophilia, confirming that at least some portion of this vast jumping gene population retains real, if usually dormant, biological consequences. A particularly striking and still only partially explained category involves so-called pseudogenes, DNA sequences that closely resemble functional genes, but that carry mutations preventing them from actually producing a working protein, remnants generally believed to represent genes that were once functional in a distant evolutionary ancestor before accumulating disabling mutations over time. Researchers have identified more than 14,000 pseudogenes scattered throughout the human genome, and while most appear to be genuinely non-functional evolutionary leftovers, a growing number of specific documented cases, published steadily through the 2010s and 2020s, have found individual pseudogenes that, despite not producing a working protein themselves, still play an active regulatory role, influencing their functional gene counterparts, blurring the once clean distinction between dead genetic material and material that retains a genuine, if indirect, biological purpose. Among the genome's most genuinely startling non-coding residents are endogenous retroviruses, DNA sequences representing the fossilized remains of ancient viral infections that inserted themselves into the genomes of distant human ancestors and have been passed down, generation after generation, ever since, now making up an estimated 8% of the entire human genome, a larger share than the roughly 1 to 2% devoted to protein-coding genes discussed earlier in this chapter. The overwhelming majority of these ancient viral remnants have been permanently disabled by accumulated mutations, but researchers made a genuinely remarkable discovery, published across several studies beginning in 2000 and led by researchers including geneticist Jean-Luc Blonde, when they found that a specific human gene called syncytin, essential for the formation of the placenta, discussed earlier in tonight's video, actually originated directly from an endogenous retrovirus, its viral fusion protein repurposed by evolution to fuse placental cells together into the specialized barrier that allows nutrient exchange between mother and fetus. The finding means a structure central to how every placental mammal, humans included, reproduces, was effectively borrowed from an ancient viral infection, a genuinely striking example of essential human biology built directly from what began as foreign genetic material, and researchers continue investigating how many other endogenous retroviral sequences scattered across the genome may have been similarly repurposed for functions not yet identified. A separate and genuinely elegant example of non-coding DNA's regulatory power involves X-IST, a long non-coding RNA gene first characterized in detail through research published during the early 1990s, responsible for one of biology's more remarkable balancing acts. Since biological females carry 2 X chromosomes while males carry only 1, female cells would produce roughly double the normal dose of every X-linked gene product unless something actively compensated, and ISTA is the specific mechanism that does, producing an RNA molecule that physically coats one entire X chromosome in each female cell early in embryonic development, permanently silencing nearly all of that chromosome's genes and equalizing gene dosage between the sexes. Which of the 2 X chromosomes gets silenced, the one inherited from the mother or the one from the father, is determined essentially at random in each individual cell. And once that random choice is made, every daughter cell descending from it inherits the identical silencing pattern, meaning adult females are, at the cellular level, genuine mosaics, patches of tissue running on a maternally inherited X chromosome interspersed with patches running on the paternal one, a phenomenon visibly demonstrable in calico cats, whose patchy coat coloring directly reflects this same random X inactivation process, though the precise molecular details of exactly how Zyst's RNA molecule physically spreads across and silences an entire chromosome remain an active subject of ongoing structural biology research. A separate and genuinely puzzling category of non-coding DNA involves what researchers call ultra-conserved elements, specific genomic sequences first systematically identified and named in a landmark 2004 paper by researchers Gil Bejarano and Davos Hussler, which found more than 480 stretches of DNA, each at least 200 bases long, that are essentially identical across humans, mice, and rats, despite these species having diverged from a common ancestor more than 70 million years ago, a degree of evolutionary preservation considerably stronger than even most protein-coding genes typically show. Since ordinary genetic mutation accumulates steadily over such vast evolutionary timescales, unless natural selection is actively removing any change that occurs, this near-perfect conservation strongly implies these specific sequences serve some function so critical that virtually any mutation to them proved lethal or reproductively disqualifying across 3 entirely separate mammalian lineages, and yet a genuinely startling 2007 follow-up study led by researcher Nadav Ahituv, in which several of these supposedly critical ultra-conserved elements were deliberately deleted from laboratory mice, found the mice developed and reproduced entirely normally with no detectable defect, a direct contradiction between the sequence's extreme evolutionary conservation and their apparent dispensability in a controlled experiment that remains, to this day, one of genetics' more genuinely unresolved puzzles. Of every unresolved question covered across tonight's entire video, none has generated more sustained philosophical and scientific disagreement than consciousness itself. The basic subjective experience of being aware of there being something it is like to be you, a phrase borrowed directly from philosopher Thomas Nagel's influential 1974 essay, questioning whether subjective experience could ever be fully captured by objective, third-person scientific description at all. Philosopher David Chalmers formalized the core difficulty in a widely cited 1995 paper distinguishing between what he called the easy problems of consciousness, explaining specific cognitive functions like attention, memory, and the ability to report on one's own mental states, all of which neuroscience has made genuine and substantial progress mapping, and what he termed the considerably harder hard problem, explaining why any of that neural processing should be accompanied by subjective experience at all, rather than occurring in principle entirely in the dark, with no accompanying felt sense of being anyone. Chalmers's formulation remains, nearly 3 decades later, the standard framework the field organizes itself around, and no proposed scientific theory has yet achieved broad, settled consensus as a genuine solution to the hard problem specifically, as opposed to further mapping of consciousness's easier, more tractable cognitive components. Among the more prominent scientific theories attempting to close this gap, integrated information theory, developed primarily by neuroscientist Giulio Tononi, the same researcher behind the sleep-related synaptic homeostasis hypothesis discussed earlier in tonight's video, and published in its most developed form beginning in 2004, proposes that consciousness arises specifically from a system's capacity to integrate information in a mathematically defined way, quantified through a formal measure. Tononi labeled phi, with systems above a certain threshold of integrated information necessarily possessing some degree of subjective experience, according to the theory, regardless of whether that system is biological or artificial. Integrated information theory generates specific, falsifiable predictions, including the controversial claim that certain simple systems could theoretically possess minimal consciousness, but critics, including a widely circulated 2023 open letter signed by more than 100 consciousness researchers, have argued the theory remains fundamentally untestable in its strongest claims and risks crossing from legitimate science into unfalsifiable metaphysics, a criticism the theory's defenders have directly disputed, leaving integrated information theory a genuinely serious but still contested scientific proposal rather than a broadly accepted solution. A competing framework, global workspace theory, developed initially by cognitive scientist Bernard Bars in his 1988 book, A Cognitive Theory of Consciousness, and considerably refined since through neuroscientific research led by Stanislas Dehaene, proposes that consciousness arises when specific information becomes broadcast widely across multiple specialized brain regions simultaneously, rather than being processed locally and separately, a model that has produced genuinely useful and experimentally testable predictions about which specific brain activity patterns correlate with conscious versus unconscious processing of the identical sensory information. Global workspace theory has accumulated considerable supporting neuroimaging evidence over the past 2 decades, but even its strongest proponents generally acknowledge it primarily explains the specific brain mechanisms correlated with conscious access to information, functioning more as a detailed map of consciousness's easy problem components than as a full explanation of why that broadcast process should be accompanied by genuine, subjective experience rather than simply occurring as unfelt information processing. The specific search for what researchers call the neural correlates of consciousness, the minimal set of neural events sufficient to produce any given conscious experience, has produced genuine and measurable scientific progressing even without resolving the deeper philosophical puzzle. Studies of patients under general anesthesia, published extensively through the 2000s and 2010s, have identified specific, measurable disruptions in long-range communication between distant brain regions, particularly between the frontal and parietal cortices, that reliably correlate with the loss of conscious awareness, findings robust enough that some anesthesiologists now use real-time monitoring of this specific connectivity pattern as a genuine clinical tool for gauging anesthesia depth during surgery, a direct practical application built entirely on a scientific foundation that still cannot fully explain at the deepest level why disrupting that connectivity extinguishes subjective experience rather than merely disrupting some more limited cognitive function. A further genuinely puzzling data point comes from split-brain patients, individuals who underwent a surgical procedure called a corpus callosotomy, severing the band of neural fibers connecting the brain's two hemispheres, historically performed as a treatment for severe, otherwise uncontrollable epilepsy. Research conducted primarily by neuroscientist Michael Gazzaniga beginning in the 1960s and continuing across subsequent decades found that in certain carefully controlled, experimental conditions, each hemisphere of a split-brain patient can process information and in some documented cases even respond to instructions, independently of the other, without the patient's verbally dominant left hemisphere having any conscious awareness of what the right hemisphere had separately perceived or done, a finding that has fueled genuine and still unresolved philosophical debate over whether such patients might, under specific conditions, house two at least partially separate streams of conscious experience within a single physical brain, a possibility with no settled scientific consensus and profound implications for what, exactly, a single unified conscious self actually requires at the neurological level. A further genuinely unsettling window into the gap between perception and conscious awareness comes from blind sight, a condition first formally documented and named by psychologist Lawrence Weisskranz in 1974, occurring in patients who have suffered damage to the brain's primary visual cortex and report total blindness in the affected portion of their visual field, and yet, when asked to guess the location or basic shape of an object placed within that supposedly blind region, perform far better than random chance would predict, all while sincerely insisting they cannot see anything there at all. Brain imaging conducted on blind sight patients has found the visual information still reaching other, older visual processing pathways in the brain that bypass the damaged primary visual cortex entirely, meaning the brain is genuinely detecting and processing the object's location and features, just without that processing ever crossing whatever specific neural threshold produces conscious visual experience, making blind sight one of the more direct and clinically documented pieces of evidence that a considerable amount of the brain's sensory processing occurs entirely outside of and independently from conscious awareness itself. Anesthesia awareness, a rare but well-documented surgical complication in which a patient remains conscious and, in the most distressing confirmed cases, able to feel pain during a procedure despite appearing outwardly and physiologically anesthetized, offers a further genuinely unsettling data point about how imperfectly modern medicine can currently detect the presence or absence of consciousness from the outside. Large-scale surveys, including a widely cited study published in 2014 by researchers at the Royal College of Anaesthetists in the United Kingdom, analyzing more than 3 million procedures, estimated the incidence at roughly 1 in 19,000 general anesthesia cases, with the standard anesthetic drugs used successfully suppressing the outward physical signs doctors have traditionally relied on to judge unconsciousness, heart rate, blood pressure, movement, even while, in these rare cases, some form of subjective awareness persisted underneath. The phenomenon has directly motivated the development and increasing clinical use of the same brain connectivity monitoring techniques discussed earlier in this chapter, giving anesthesiologists a more direct physiological readout of awareness than outward physical signs alone can provide, though no monitoring technology currently available has achieved perfect, universally reliable accuracy, leaving the reliable, real-time detection of consciousness itself, even under tightly controlled clinical conditions, a genuinely unsolved technical problem. A further foundational puzzle in consciousness research, called the binding problem, addresses a question so basic it's easy to overlook entirely. The brain processes an object's color, motion, shape, and location using genuinely separate, physically distinct neural pathways, each specialized for its own specific feature and each operating with its own slight processing delay. And yet, a person perceives all of these separately processed features unified into one single coherent object experienced in a single seamless moment, rather than as a scattered collection of disconnected sensory fragments. Neuroscientist Wolf Singer's influential research, published beginning in the 1990s, proposed that the brain solves this binding problem through neural synchrony with the separate groups of neurons processing an object's different features firing in precisely coordinated, synchronized rhythmic patterns, effectively tagging themselves as belonging to the same object through shared timing alone, rather than through any physical connection between the regions involved. Singer's synchrony hypothesis has accumulated meaningful experimental support in the decades since, but it remains genuinely contested as a complete solution, with several competing theories proposing the binding might instead happen further downstream, in specific integrating brain regions, and no single account has yet achieved the kind of broad, settled consensus that would let neuroscientists say the binding problem, like the closely related hard problem discussed above, has actually been solved. At the very tip of every chromosome inside nearly every cell in the human body sits a structure called a telomere, a repeating, non-coding DNA sequence that functions much like the plastic cap at the end of a shoelace, protecting the chromosome's genetically vital core from fraying or degrading during the routine process of cell division. And the specific, measurable way telomeres shorten over a lifetime has become one of the most closely studied and, still only partially explained, biological clocks connected to human aging. Each time a typical human cell divides, its telomeres shorten slightly, a consequence of a specific limitation in how the cellular machinery responsible for copying DNA physically works, first mechanistically explained through research conducted by molecular biologist Elizabeth Blackburn, biochemist Carol Greider, and molecular biologist Jack Szostak, whose combined work identifying telomeres and enzyme telomerase, which can rebuild shortened telomeres under specific conditions, earned all three researchers the 2009 Nobel Prize in Physiology or Medicine. Once a cell's telomeres shorten past a critical threshold, the cell typically enters a permanent non-dividing state called senescence, or undergoes programmed cell death entirely, a built-in limit on cellular division first empirically documented by biologist Leonard Hayflick in 1961 and now formally known as the Hayflick limit, a discovery that overturned an earlier, widely held scientific assumption that ordinary human cells could in principle divide indefinitely. Telomerase, the enzyme capable of rebuilding shortened telomeres, remains active at meaningful levels in only a limited subset of human cells, including reproductive cells, certain stem cells, and, significantly, an estimated 85 to 90% of all cancer cells, according to figures compiled across multiple published cancer studies. a connection that has made telomerase itself a genuinely significant and actively pursued target for new cancer treatments, since a cancer cell's ability to continuously rebuild its telomeres is a major reason it can divide indefinitely, bypassing the Hayflick limit that would otherwise eventually halt an ordinary cell's runaway proliferation. This same connection creates a genuinely difficult biological tension that aging researchers still haven't fully resolved. Artificially boosting telomerase activity throughout the body might, in principle, slow or partially reverse some visible signs of cellular aging, but doing so would also meaningfully increase the risk of uncontrolled cell division and cancer, meaning telomere length cannot simply be treated as a dial to turn up without carrying a genuine, well-documented cost. A landmark 2004 study conducted by health psychologist Elissa Epple and Elizabeth Blackburn found that mothers caring for chronically ill children, a group experiencing sustained, measurable psychological stress, showed significantly shorter telomeres than mothers of healthy children, with the effect scaling directly with the number of years spent as a caregiver, one of the first studies to provide direct biological evidence linking chronic psychological stress to accelerated cellular aging at the molecular level, a finding replicated and extended across numerous subsequent studies examining other forms of chronic stress, though researchers still debate exactly how much of an individual's overall telomere shortening across a lifetime be attributed to psychological stress specifically, as opposed to the many other genetic and environmental factors known to influence the same underlying process. Beyond telomere shortening, aging researchers have identified several other measurable biological changes that accumulate with age, including a phenomenon called epigenetic drift, gradual shifts in which genes get chemically switched on or off over a lifetime, without any change to the underlying DNA sequence itself. First comprehensively measured and formalized into a predictive epigenetic clock by biostatistician Steve Horvath in a widely cited 2013 study, which found that specific, measurable chemical modification patterns across roughly 350 sites in the human genome could predict an individual's chronological age with remarkable accuracy, often within a few years, across virtually any human tissue type tested. Genuinely more significant to aging researchers than the clock's raw predictive accuracy is the finding that a person's epigenetic age, as measured by Horvath's clock, sometimes diverges meaningfully from their actual chronological age, with individuals whose epigenetic age runs faster than their calendar age showing measurably elevated risk for age-related disease and earlier mortality. In multiple follow-up studies, suggesting the clock may be capturing something biologically meaningful about the pace of aging itself, distinct from simple calendar time, though precisely which specific lifestyle and environmental factors most directly drive this divergence, and by exactly how much, remains an active and only partially resolved area of ongoing research. A small population of centenarians and supercentenarians, people living past 100 and 110 years respectively, has drawn particular scientific interest specifically because many show a genuinely puzzling pattern. They often carry the same disease risk genetic variants found throughout the general population, including variants linked to heart disease and cancer, yet somehow avoid developing the associated diseases at anything close to the expected rate. Research conducted on concentrated longevity populations, including a widely studied group on the Italian island of Sardinia, has identified several candidate protective genetic variants that appear more common among centenarians than the general population, but no single longevity gene has been confirmed as the primary explanation, and most current longevity researchers believe extreme human lifespan likely results from a genuinely complex combination of multiple modest effect genetic variants working together with lifestyle and environmental factors rather than any single clean genetic switch waiting to be discovered. The naked mole rat, a burrowing rodent native to East Africa, has become one of aging research's most closely studied and genuinely puzzling animal models, specifically because it appears to defy the normal biological rules of aging almost entirely. Unlike similarly sized rodents, which typically live only 3 to 4 years, naked mole rats routinely live past 30 years in captivity while showing almost no measurable increase in mortality risk or decline in reproductive capacity as they age. A pattern researcher, Rochelle Buffenstein, who has studied the species since the 1980s, has described as negligible senescence, essentially aging that doesn't follow the standard trajectory found in nearly every other studied mammal. Naked mole rats also show a strikingly low cancer incidence, and a 2013 study led by researcher Vera Gorbanova identified an unusual sugar-based compound called high molecular mass hyaluronan present in unusually high concentrations in the animal's tissue, which appears to make its cells resistant to the crowding that normally triggers uncontrolled tumor growth, though researchers still don't have a single unified explanation connecting the naked mole rat's cancer resistance, its unusual longevity, and its apparent immunity to normal cellular aging into one confirmed underlying mechanism, leaving the animal a genuinely active subject of ongoing aging research rather than a fully solved case study. Separately, caloric restriction, deliberately reducing food intake below normal levels without causing malnutrition, has been shown to measurably extend lifespan in a wide range of species, from yeast to worms to mice, in research accumulating since a foundational 1935 study by nutritionists Clive Mackay and Mary Crowell first documented the effect in laboratory rats. The precise mechanism has been partially traced to a cellular pathway called mTOR, which senses nutrient availability and, when suppressed by reduced food intake, appears to shift cells toward increased maintenance and repair activity rather than growth. But a large, long-running study on rhesus monkeys conducted by the National Institute on Aging, publishing results in 2012, found a smaller and more ambiguous lifespan benefit than earlier rodent studies had suggested, complicating the picture considerably for how directly the effect translates to longer-lived primates, and leaving the practical relevance of caloric restriction for extending human lifespan specifically as a genuinely open question rather than an established fact. A rare genetic condition called Hutchinson-Gilford progeria syndrome, affecting an estimated 1 in every 4 to 8 million births, offers researchers a genuinely rare and clinically significant natural window into the aging process, since children born with the condition develops symptoms strikingly resembling accelerated aging, including hair loss, joint stiffness, and cardiovascular disease, typically dying of heart attack or stroke around age 14, decades earlier than the same conditions would normally appear. The specific genetic cause was identified in 2003 by a research team led by geneticist Francis Collins, who found a single mutation in a gene called LMNA, responsible for producing a protein that provides structural support to the nucleus of every cell. And the mutation causes a malformed version of the protein, nicknamed progerin, to gradually accumulate and destabilize the nuclear structure over time. Genuinely striking to aging researchers is the subsequent finding, published through the 2010s, that low levels of the same progerin protein also accumulate naturally in ordinary aging cells from people without progeria at all, suggesting the same specific molecular process driving this rare and severe pediatric disease may also play some smaller, ordinary role in typical human aging, a connection that has made progeria research a genuinely significant, if still incomplete, window into understanding the aging process more broadly, and that led directly to the first FDA-approved treatment for progeria, a drug called lonafarnib, approved in 2020, which modestly extends patients' lifespan without fully halting the disease's progression. A second and genuinely distinct accelerated aging condition, Werner syndrome, offers researchers a useful point of comparison, specifically because it strikes considerably later than Hutchinson-Gilford progeria, typically leaving patients to develop entirely normally through childhood and adolescence before symptoms resembling rapid aging, including cataracts, skin changes, and cardiovascular disease, begin appearing in their 20s, with affected individuals often dying in their 40s or 50s from age-related conditions arriving decades ahead of schedule. The condition was traced in 1996 by a research team led by geneticist Gerard Schellenberg to mutations in a gene called WRN, which produces a protein essential for properly unwinding and repairing DNA during cell division, a genuinely different molecular mechanism from the structural nuclear protein defect responsible for Hutchinson-Gilford progeria, and the existence of two distinct genetic pathways that each independently produce a recognizable, accelerated version of normal human aging, has become a genuinely significant piece of evidence for aging researchers, suggesting the aging process itself may not trace back to any single root cause, but instead emerges from the accumulated breakdown of several separate, independently essential cellular maintenance systems, each capable, when specifically disabled, of producing its own distinct flavor of premature old age. We've moved tonight through 10 chapters and 200 facts about a body every listener has lived inside every single day of their life, from the still unexplained evolutionary purpose of a third of that life spent unconscious, through the vivid, half-understood narratives the sleeping brain generates every single night, into the vast independent nervous system quietly running your digestion, through the phantom pain a missing limb can somehow still produce, past the involuntary blush and the goosebumps. Evolution never bothered to fully retire, through an immune system that somehow remembers a threat from decades ago, into the 98% of your own genetic code that resists any simple, tidy label, past the single hardest open question in all of science regarding your own subjective experience of being anyone at all, and finally into the quiet cellular clock ticking at the tip of every chromosome in your body right now. It's worth sitting in these last few unhurried minutes with just how strange that overall pattern actually is. You are, at this very moment, the most thoroughly studied object of scientific inquiry that has ever existed, examined across medicine, biology, chemistry, physics, and psychology for longer and with more accumulated resources than any other subject in the history of organized human research. And yet the specific body carrying you through this sentence still keeps secrets from the very field of science built specifically to understand it. Not because anyone has been careless or incurious, but because a system this layered, this many billions of years in the making, was never going to yield every one of its mechanisms easily, or all at once, or on any convenient human timeline. Some of tonight's 200 open questions will likely find clearer answers within the coming decade, as tools like single-cell genetic sequencing, increasingly sophisticated brain imaging, and the kind of large-scale microbiome research, still in its comparatively early stages, continue maturing. Others, especially the hard problem of consciousness, sitting at the center of Chapter 8, may remain genuinely unresolved for a very long time yet, quite possibly for longer than the working career of any scientist currently investigating it, a genuine and honest possibility every serious researcher in that specific field has had to make some kind of peace with. That unresolved quality was never really the discouraging part of tonight's video, if you think about it clearly for a moment before you let go of the thought entirely. Your own heart has been beating, your own gut bacteria quietly working, your own immune system quietly remembering every single day of your life, whether or not anyone, including you, ever paused to ask exactly how. The not fully understood parts of your own body have been carrying you perfectly well regardless, tonight included, and they'll keep doing exactly that while you finally stop asking any questions about them at all. Somewhere behind your own closed eyes right now, your suprachiasmatic nucleus is already quietly working toward the sleep. This whole video was built to help along. Your telomeres are exactly the length they are without needing your permission or your attention. And whatever the hard problem of consciousness ultimately turns out to mean, there is undeniably something it is like to be you, lying here, letting 200 small mysteries settle instead of solving them. Nobody in any laboratory anywhere is waiting on you tonight to figure out why you dream, or why you blush, or what that last 2% of your own genome is quietly doing while you're not looking. That work belongs to people who will still be doing it long after tonight, in the morning, and for as long as bodies like yours keep being interesting enough to study, which, on the evidence of everything covered so far, looks like it will be for a very long time yet. So let this be the last one. Let the body that's been running itself patiently and mostly invisibly for your entire life keep doing precisely that, the same quiet, unexplained work it always does. Once the lights go off and the questions finally stop, and let all 200 facts you've just heard settle now, gently, into the kind of rest that never once needed you to understand it first.